It's no secret that reading scientific journal papers is hard. The topics are highly technical, the language is often intentionally obtuse, and the subjects are very niche. But also, there are so many amazing papers on topics you probably never knew existed. So we created Journal Club. It's a live hangout on the new SciHouse Discord where once a week someone signs up to present a paper they're interested in and break it down piece by piece. Everyone gets a chance to learn something new, and it's a really fun way to learn the skills of reading papers. So if you want to join in, there's links below. Now, without further ado, this is Journal Club.
Nice.
Yeah.
Did anybody have a chance to actually look at the paper?
Yeah.
Just cool, because I was thinking that I have some notes here. I'm just pulling up the paper. Give me a second. All right, the paper is kind of dense, and so I thought it would be a good idea to go over somatic embryogenesis simultaneously with showing how the paper is going over it, because while it's a really good paper and a really good example of a review paper, it kind of blurs some of the steps that I find this to be really common in the SE papers, that they just kind of mix up a few of the steps and it's not super clear. So I think it would make sense because the idea is not that complicated. Hello to everybody just joining us. Yeah. All right. So I'm gonna start. Is that cool?
Mm-hmm. Let's do this.
Right. So I know a lot of people here because I've been talking a lot about syn seeds, and synthetic seeds are super cool concept because they beat out seeds in a lot of ways, and of course they have shortcomings. But this is a really important paper if you want to get into syn seeds, because it shows you what's at the heart of syn seed and how are you able to put a little piece of a plant inside some media and have it grow into a new plant. Because some plants it's really easy, like potatoes. If you put a piece of potato in something, and you can kind of get an idea of it. But for most plants, you can't just get a piece of it and expect it to grow. So what we're doing here is called somatic embryogenesis, which is a great word. I just want to underline that. I love saying it. So somatic embryogenesis is basically, you guys know what an embryo is. Embryogenesis is making an embryo. Usually this just happens in a seed. So you have this female cell. I think it's called a megaspore, and then the pollen comes in and it double penetrates the megaspore and gives it 2 nuclei, and then all this complicated stuff happens, and you end up with this cell that grows next to a lot of fuel, which is the cotyledon. Oh, sorry, not the cotyledon, the endosperm, and that's its fuel. And that's a normal seed, and that can grow into a whole new plant. It's ready for it. It knows when to time the right hormones to be like, okay, grow some roots. Okay, grow some leaves, and it'll do all this on its own. So we're basically trying to get the cell of a plant and confusing it and be like, hey, I know you were a leaf cell. In actual fact, we tend to use the inside of a seed, but you can use any cell technically. You get the cell that knows what it is, and you make it forget what it was. You confuse it, so you go back to an embryonic state. And once it's in that embryonic state, and we do that by just kind of replicating the hormones that would have normally been inside the seed, then it just thinks it's a seed again. And then with the right environmental things, it'll start developing like a seed would. So we give it a little bit of fuel, a little bit of hormones, package it up in a nice gel cap, and we have a synthetic seed. That's the basic idea of it. So we're going to go over this paper. And I know Gabe talked last time about his paper and what a good structure of a paper was with the fishtail at the beginning and the end, and how the paper that he showed didn't actually follow that structure. And it is a great model, and a lot of papers do follow it, but here's another paper that doesn't quite follow that, because this is a review article. And review articles are really cool to get into because they're a great starting point for a lot of things. They're basically people that analyzed a whole bunch of other studies and told you what they found in general and maybe point out some trends that are happening over the years. Anyway, sometimes they can be really shitty because sometimes the people just didn't want to do very much work, so they decided to do a review. But sometimes, like this paper, you have really cool stuff, and it's somebody who really tried to be on the cutting edge of what that field was. And that's what we have in this paper. I think it's 2019, and it's pretty modern for 2019. It's been about 20, 30 years since they started doing somatic embryogenesis. And so there's a lot of backlog of things that kind of work, things that didn't work, promising leads. So this is a good one to start with. So it starts out with just an introduction and kind of why they're doing the paper, why it's important. I have some highlights here that just kind of tell you in this first part, which is Introduction to Somatic Embryogenesis. And it's basically saying why this is important. It's important because making 1,000,000 plants is really hard. You know, if you want to get 100 plants, okay, it's not a big deal. I can go to a nursery or something, maybe even like 500 plants. If I'm here in Bali, right, and I want to plant a farm, if I want 1,000 plants, that's kind of hard. Like I have to basically clean out the whole island's worth of nurseries to get that. But if you want like 20,000, 100,000, 1,000,000 plants, you have to do micropropagation. And when you're doing that, it's so much cut and paste. Like it's just you or a bunch of other people in tandem cutting little pieces of a plate and moving it to other ones. It still takes a long time. So the reason this is important is because instead of all that little cutting and pasting on little petri dishes, we can just make a smoothie, and a magic smoothie that turns into 1,000,000 little plants. And like the cell densities are actually pretty high. So theoretically, you could have a liter of suspension culture, which you turn into embryos. And then if you were able to separate those out, like you would easily have hundreds of thousands of plants. Of course, there are limiting factors, but it's still the fastest way to make a lot of plants. In 50 years, whatever plants and farming is like then, I somehow doubt they're going to get more efficient than a liquid suspension culture of a bunch of plant babies. It's just so much. So yeah, and then so these are the basic stages. What do you do once you get this plant cell? And here it lists them out as initiation, multiplication. Let me find a good way to scroll this with a half screen. Hold on a second. Okay, yeah, we got initiation, multiplication, and maturation, and then germination and plant formation, which all rhyme, which is actually kind of confusing now that I say it out loud. Yeah, so you could read through these. I have some highlighted things that kind of show the best parts of these, but I'm actually going to go over my own version of those 3 steps because I think it should actually be like 4 steps, and they're just mushing 2 together, and it's really confusing. So basically the steps that they're actually trying to address, and they're trying to automate these steps. So they're going to analyze what are the steps, and then they're going to look at each of these steps and be like, can we make this better? Is there a way we can make this faster? Can a robot do this? Is there any reason that we're doing it very manually and very space intensively? So first it has to analyze what it has to improve. So the initiation step is actually like 2 steps. It's basically first you take the piece of the plant called an explant. We can talk about that later. You can get it from a bunch of different parts of the plant. It turns out that some parts of the plant work much better for this than others. So let's say you've already got the ideal part of the plant. You got it. Next thing you got to do is you got this little pack of cells, and you need to make a lot of them because you need to make a lot of cells to put into your liquid. So you multiply them. The easiest way to do this is with a callus culture. The callus is basically you give it too much of every hormone, and it just ends up making like a giant lump of cells, and they just continue to grow as a lump, which is great because it lets you take off some whenever you want to try them out. You can actually take the plant and directly induce embryogenesis in the plant, but you don't get very many cells. Like you only had as many cells as you cut out of the plant initially. So while that's cool and faster, it doesn't actually help us for our purposes, which is making a fuckload of plants. So you have this little plant booger growing in your dish, and it's just 1,000,000 little cells. Some parts of that callus are going to be bright green. These are called PEMs, pro-embryonic masses. But they just mean that's the good stuff. That's the stuff that's going to turn into little baby plants. So we cut that out, and we put it into a liquid. And in that liquid, this liquid has a lot of auxins. And these auxins and a few other thingies are basically there to replicate that initial stage that a seed undergoes right after it's been fertilized, because all these hormones start being released, and the plant's like, hey, little zygote, like wake up, you gotta do your thing and start replicating. So we're basically doing that right now. But now we have, instead of 1 little cell is getting confused, we're confusing hopefully 1,000,000 little cells, and not all of them will be sufficiently confused. Some of them have a lot of gumption, and they hang on, and they're not really useful. But if we're clever and we use the right hormones and stuff, we can have a good amount of them become embryonic. So then in the same step, you do the induction rich meter, which is you really increase that auxin, and that's what actually turns the PEMs into somatic embryos. And now, congratulations, you have a bunch of somatic embryos. They have a very particular shape. You can look up the names of the shapes as these things develop. This one's called globular. It just looks like a blob. I think it's like 16 cells or something.
When you say this one, can you point to where on the document you saw the
Yeah, is there a way to like project my screen or something so I can
Yeah,
play
you
stuff?
can do a screen share, and you can also just you can say
All
like
right, all right.
page 4
All right,
for
cool.
those of us following along at home.
I know you guys are just looking at my face, so it makes more sense to look at the paper. Let me figure out how to do this.
But I mean, now we
Okay.
should also have. Hopefully everybody has the paper up, right?
Yeah, it's cool. It's just easier for me to talk about, and I can like point to it with my arrow key.
Yeah, there's a big screen button.
Yeah, I see it. Okay. Oh, I gotta pick what I'm streaming. Okay. Sorry, I was saying earlier, this is the first time I've actually ever done a group video voice chat. I don't know the like just how any of the signals.
The weirding. You haven't learned the weirding ways.
No, I haven't, man. I've been in the jungle too long. I got my own weird going on. All right, what do you guys see? Do you guys see me?
We can see the Discord.
We're good.
You see my desktop?
Yep.
Okay, dope. All right. Yeah, so we just went over initiation, which is actually mushing together the initiation and multiplication. They just make it really confusing because you got to make a lot of cells before
make the paper full screen. We don't need to. We already see us. Yeah,
How's
we want to
that?
see that. There we go.
Don't see anyone?
Is that cool? Can you guys all see that? Hello?
The resolution is kind of low.
Okay, let me try. Aha. All right.
There we
Is
go.
that better?
That's really easy to read now.
Yeah? All right, cool.
Yeah. That
All right.
4 HD science journal action.
Yeah, but I'm still not used to having decent internet. It's a very, like hot water, it's a very precious thing that I'm still in awe of. Okay, so we went over the initiation and multiplication. I think this paper kind of mushes them together. But you have a good idea. We made a lot of cells, and then we convinced the good ones to become embryos. So this is one of the stages where it's that multiplication stage where you're making a lot of cells. And that's one of the first places that you can really use automation. So you can grow a callus culture in a solid media, like on agar. But it turns out you could also grow that, and this is particularly useful in conifers, you can also grow that callus floating in a liquid, in liquid media. And you're putting in like a certain amount of enzymes and a little bit of movement in there so that the cells break apart. And so they actually grow faster that way because every cell is kind of cleaving off and splitting into more cells instead of you having to cut it manually or growing like a blob. And when it grows like yeah.
Is this kind of like making plant organoids?
So organogenesis is a little different than this because organogenesis is bipolar and this is monopolar. And so for organogenesis, you have to do different phases of hormonal interactions with the plants in order to get to do what you want. Somatic embryogenesis is great because it just needs one hormonal cue, and that's like, it's different, but it's based on auxins and stuff. And so it's a little, yeah. That makes sense.
We're using this term differently. In mammalian cell culture, normally your cells are attached to a plate, but you can also make organoids, which are cells that are floating in media that create their own little boogers, as you term them.
This is different. This is more like plant smoothie. Like, so if you take a callus and you dump it in media and you keep the media moving, it literally just makes soup. Like, it makes like living green soup. Like, it doesn't clump. You got to get the movement speed right. But yeah, it doesn't clump. It only clumps when you stop it moving.
Yeah, they want to clump naturally, but you don't let them. That's the point of keeping them in this culture. Does that answer your question, Gabe?
Yes, it does. Thank you.
All right, cool. Yeah, so basically it's saying here that, this paper is focusing on conifers, which are super cool plants. I actually don't have that many of them here, except I was showing you guys like up in the tall mountains, like over 1,000 meters, and you start to get some podocarpus. But so this is interesting that there's a difference between angiosperms and conifers in that angiosperms, at least most of the time.
Explain that for people that you got to remember, we got a lot of different skill levels.
Yeah, sorry guys. Angiosperms are plants that make flowers and fruit. Conifers are things with needles and cones, generally. So that's pretty much it. They kind of have different branches, and they, like, down on a functional level, they're behaving really differently. So this paper kind of reveals that when you have the embryos of their cells, they have different requirements. The difference being that it seems conifers much prefer to have like a solid surface that they're sitting on, at least sometimes, whereas a lot of angiosperm, apples, carrots, most of the plants that you would think of that aren't piney are probably angiosperms. A lot of them can get by on just pure liquid, so they don't actually ever need to rest on a solid surface. And so this stage right here, where it's referring to the different multiplication steps, it just kind of goes over that. It goes over, hey, look, we should, basically, you kind of got to flood and drain the little cells of the conifers. I'm sure you guys all like hydroponic kind of stuff, because it still gives them the solid surface, but when you flood it, they're getting all the nutrients they ever need. Because remember, what is the difference then between these little embryos that we just made and the embryos, the zygotic embryos that live inside of the seed? The difference is we have to give them their food. This is just a naked little embryo. It's like normally they have the equivalent of an umbilical cord. I think it's called a tube something. And it connects to the food, the stuff that's in the seed, the white stuff. It's either starches or oils or something. And that's what gives a little baby plant fuel to start making leaves, so it can start making photosynthesis stuff. And it doesn't have this. It's, what'd you say?
Making a humidified.
Humidified? I'm not sure.
All right, like I was just.
Yeah. So.
Yeah.
What was I at? So. Yeah. So we're just trying to see. Hold on. That's sorry. I got to go plug in my computer.
So hey, since we're in limbo, does anyone else want to talk about the paper?
I'd love to, but it's outside of my league at this point. I still, I have read it multiple times, but I still gotta, yeah.
Well, I mean, I remember working in labs with people who were working on their PhDs, and they would spend half of their day just reading the same paper over and over and over again.
Yeah.
So don't worry about it.
Right.
So
but awesome concept though. Like definitely, it basically sums up like the you and Justin talked about like how to induce like totipotency and is it yeah. Well, this definitely
technically totipotent, but
yeah, totipotent. Okay,
yeah.
Okay, yeah. Pluripotent are the stem cell we got.
But so this concept of totipotent isn't one-to-one when it comes to plants. Like when you say inducing, it's not like mammalian cells where you have to literally genetically modify them to do it. But all plants just do this. Like every cell in every plant can just do this.
Kind of ambiently is a totipotent cell.
Yeah, like if you give them the right hormones, they'll do whatever you want them to do.
Or you don't even have to give them the right hormones. Like
if you're doing culture, you absolutely.
If you're doing culture, but like also when it's alive, like so I have this tomato plant. It's a volunteer tomato plant. It decided to grow and become this giant monster in my backyard, which I am pro giant monster. And then Bost was like, hey, what are these like little white bumps that are coming out of the part that's touching the ground, because we lifted it off the ground, and I'm like, oh, that's where it decided to just become roots. Hey, he's back. So anyway, the point is that any cell in a plant is ready and willing with the right triggers to become another type of cell. That's the best part. Yeah.
Right.
So,
I mean, like in trees, for example, there's this concept, especially if in bonsai, there's this concept of it's called air layering, where basically you want roots to show up way up on the tree. So like if you've got like a big thick branch, so if you want to make an itty bitty bonsai but you want a big thick trunk, the easiest way to do this is to go to an old tree and you basically cut a ring of the bark off of one of the big branches, and then you like coat it in sphagnum moss or dirt or whatever, and the plant basically panics because you've cut off its ability to get nutrients and water, and so it as long as you keep that area moist, it just starts putting new roots in. So like it doesn't care. Like plants are just to steal a line from a friend of mine, plants are basically just like hormone distributions over space and time. Like you give them the right hormones in the right area, and they just do the thing. Or if you want them to grow new branches, you cut off what's called the apical meristem, which is basically the bit on the very end where all the hormones are being produced. Because a lot of like leaf growth or branch growth is triggered when a certain hormone level gets too low. So basically, the way this works is when the apical meristem has grown enough, it is literally far enough up the thing that the hormone doesn't travel very far. So once it passes a certain point, the plant knows, okay, I need a new leaf here. But if you just want more leaves there, you just cut the meristem off, and it goes, oh, the hormone's low, and it starts making new branches.
Diego is back. Diego's back, and God only knows how long we'll have him again. So back to Journal Club.
There's only one planet in between us. It's not that much.
Yo, Artem, your mic is fucked.
Okay, can you guys hear me okay?
Yeah,
we can hear you. It's whenever Artem talks, it sounds like somebody put a robot in a blender.
Yeah, definitely. That was kind of weird. Yeah, so I mean, what you're saying was kind of cool because we're talking about some, somatic embryogenesis and using those in synseeds. But there are other parts of the plant that can also be used, just because, like you said, it's just up to the hormones, and other parts of the plant are available. So, like another very viable technique for synthetic seeds is using shoot tips, either from like multiplication or like just literally cutting the tip off some plants and then sticking them into a synseed. Yeah, so I mean, it's pretty cool how variable it can be. But there are some like that are the most reliable parts of the plant that are just like the most open to being convinced to be something else. And we'll go over at the end, like, what are the because why can't we just why isn't everybody doing this? Why isn't it happening for all the plants? Like, what are the limiting factors? And some of those are like, yeah, I mean, you can just throw hormones at it and convince it to do stuff, but there's a lot of variability on how it responds to that, depending on like 5 or 6 factors. And one of those factors, as always, is just electrocuting the fuck out of it, and sometimes that works. But you can do it the fancy way more often. So where were we last time? We were talking about some of the automation steps here with the conifers, and yeah, how you basically make this smoothie. And this smoothie is a key part to really cranking up the volume on this thing, because this is all a lot of work if you're not making a lot of plants. If you're gonna make just a few hundred or a few thousand plants, just do normal micropropagation or make seeds and cuttings. Like, it's not that hard. It's just when you get to the upper reaches that you're like, oh man, I wish we didn't have to have a whole factory worth of tissue cloning people. And that's what this is trying to address. So it's important to think about this in scale, not like, oh, that's like a lot of work for a few plant embryos. Ideally, you should be able to scale this up. Here we have some really pretty pictures, and we were talking about some of the different stages that the somatic embryo goes. They have good names like globule and torpedo and heart shape and stuff like that. Yeah, and just basically different stages are going to behave differently. So ideally, for this application, you want them to be stage synchronized, meaning that they're all at the same stage, and they don't all want to do that. They'll respond a little differently just from natural genetic variation, because you just made them have a million replications. And so there's clever techniques that you can do, either physical or chemical, that'll let you find the ones that are either like all torpedo stage or all heart shape stage. Okay. Yeah, so basically, after you have made a lot of them and you've turned them into embryos, you got to tell them, all right, embryos, do your thing. Like, start turning into a plant now. And then you take them out, you wash the hormones that you had on there before, and you give them totally new hormones with almost no auxin. Although sometimes they might need a tiny bit of auxin, but pretty much no auxin. Basically, so now that makes them start maturing. And so we call this the maturation step. And this is when they're going to start to develop, if they do develop, because remember, some of them just don't. If they do develop, they'll follow these stages that you see in B, C, D. And yeah, so now you're going over a part here. Like, how do you make them do this? When they're starting to mature, what are their requirements? Because they're not the same that they were to multiply them, nor are they the same to turn them into into embryos. The requirements are a little different. Also, by the way, this is important. They don't divide anymore. You're not making any more of them. So whatever you had to start with is the ones that you're going to have to end with now. The stage to make the cells is the multiplication stage. There's no other part where they're like growing and dividing. In fact, that's a bad thing. If it happens, it means they're not synchronized and stuff is going on. Yeah, so basically, how do you do this? Well, you need to put them on something solid, usually. Sometimes you don't have to, but to be safe, we'll say that you put them on a solid surface. The study kind of goes over a bunch of different methods for that. Some of them put them on like a semi-solid medium, like a sponge with nutrients in it. Other ones did the flood and drain. Some of them did like a gel thing. There's a really cool list here at the end that kind of compares all these. So this is just kind of going over the different things that people have tried and which ones are typically the most successful. And the last stage is germination and plant formation. All right, so we got a bunch of these little guys. We separated them out, and they look like here. Everybody can see my screen, right? Yeah?
I cannot.
No?
I can.
Negative.
Oh, okay.
Hey, click on the square that shows the screen. People.
It's there.
I don't see.
It's working
a
fine.
square.
That dude's just doing it wrong.
I don't see a square that shows.
Click on voice and video chat. It will show you a grid.
I mean, if you just click next to Diego's name, just click the thing that says live. It'll take you to it.
But if Discord is taking a dump like it used to, maybe you just restart Discord.
Should be fine.
Yeah? Are we there yet?
Yep.
Okay, as you were saying. Also, you could just look at the paper. It's page 4.
Yeah, just look at the paper. That's a good idea. With the prettiest pictures in the paper is kind of what we're on now because it shows all the stages.
Yeah. Page 4,
so,
people.
Yeah, God, I've never said pages out loud. Okay, so here it's actually, I've kind of gone over what it's saying here because the next part, oh, sorry, I skipped germination and plant formation. Yeah, so you got these little embryos, this stage E right here, and now they're ready to go. So you just got, if you put them in the right place, they would do their thing. In fact, you can even put them directly in like soil media and they'll grow. Not every time, and they're quite delicate, but it's possible. Just like all the ingredients are there. But they also need a few other things. So typically what would happen in a seed is that the zygote starts to be like, all right, I'm done getting ready. Now it's time to get into seed mode in case somebody wants to eat me or something. And so it's got to dry out a little bit and not just be a bunch of wet tissue. And so they'll start to release certain proteins and desiccate a little bit. And this kind of triggers hormones, and then it's like ready. Depending on where the seed is made, tropical or temperate, it can be dormant. Like some seeds just won't sprout for a year. Other ones don't. Doesn't matter. We don't have that. Like we don't have the desiccation step. We can imitate it a little bit, and we can actually even add in some of those hormones that would normally be present in the seed to help. It helps. So it helps the seed along. But there's still like glaring differences in how this embryo is going to behave compared to a seed. Yeah, I mean, I tried to write down some of the stuff here, some of the differences between them, but it's like essentially, they just don't have a strong connection to like a fuel, and so they're really delicate in that way. And also, okay, yeah, and they also don't sprout as much as seeds. So like, if you have 5 seeds and 5 syn seeds with somatic embryos, more of the normal seeds are going to sprout. We can make up for this because it's a lot easier to make a lot more syn seeds, but that's still like what we're dealing with. They're just not as sturdy as a normal seed.
But also, you and I talked a little bit about chitosan, right? I know.
Yeah.
Sorry to bring it up. But...
Yeah. No, definitely. So like how you make these syn seeds and how do you make the somatic embryos more likely to survive? That's a huge issue. Like that's super important. And it's gonna, every time you make a few % increase, you've multiplied your efforts instead of just making double the seeds. But there is a give and take, right? Because sometimes it's easier to make twice as many of a super easy cheap seed than it is to make, half as many of a very productive seed. So you got to like figure out what you're doing and exactly what the effect is, I think. But chitin's a really good one, I think, because it's antibiotic. A lot of this is because you're just putting sugar around this thing. So if a bacteria gets in there, it can just eat the sugar and destroy the embryo before the embryo even has time to use it. This doesn't happen in seeds because they have a lot of protective measures, usually. But that's one of the major things they have to deal with. And we'll actually see right here, a major unresolved problem with artificial seeds is woody species have been low. And that's because of bacterial contamination of the fluid. If you're doing it small scale, I mean, just put a little bit of antibiotic, but if it's any kind of scale, like you can't do that. So you need something like chitin or there's a bunch of other options too. We can talk about that. There's a lot of, this is such a big paper. This is the kind of paper, and the references are great. So like if you dig into each of these references, there's really cool stuff in there. This big chart where they list out everybody's species and what they tried to do with suspension culture and somatic embryogenesis. Super cool. I mean, I've been digging through this, and like this is definitely just a first glance over the paper. What are they trying to do? How are they trying to do it? And where are they pulling from? Because where are they pulling from is super important, and you're expected to like dig through it after a while. I mean, that's what I'm still doing. Yeah, so we've kind of been over the parts that can be automated. Of course, are the liquid part, and the other part, which I actually didn't say, so that was too bad. But it's when you are trying to identify which are the somatic embryos that were successful and which ones aren't. So there's a lot of ways to do that. It seems like one of the most successful ways is to plop them out onto their media, either like a sponge or something, and then just point a camera at them, teach the camera what a good one looks like, and pick that one up and use it. So that's cool, and it's pretty low tech, but it's a lot of programming. There's also chemical ways to do it, but going over them, I'm sometimes a little dubious about how scalable it is. It might be the most scalable thing is just to automate the shit out of a camera with a robot hand attached to it. That's what this paper is saying is a really good way. And they have this really nifty picture of what a setup actually looks like. And I would absolutely love to just hang out and watch a bunch of plantlets pop out. And there's a good one in India, and my friend lives near there. And so we actually want to go and check out their facilities, because their goal is like, right now they're doing 1 million plants per year via this method. And their goal is 10 million for reforestation purposes, which is so many plants. And I would really like to see just like what the daily operations of that are. Okay, this is pretty much kind of finishes up, has some concluding remarks and like state of art and perspectives in the future. I'm just gonna go over really quickly, and then I can like answer questions. Why these things are better than seeds? Because I think that needs to be asked. So just really quickly, if you're trying to, I told you if I need a lot of plants, use this kind of thing. But also there's other things. Okay, you guys, or a lot of places have these things called seasons. So there's just, I need 100,000 plants. Where am I going to get the seeds from? Well, can they time them all? So like all the plants don't flower at the same time. So how do I get the seeds? Some seeds, especially tropical seeds, don't last very long. We don't have a dormancy period, so things sprout immediately. And if they don't do that, there's much less success rate. So, okay, so now we can produce any kind of plant at any rate at the same time as other ones. So we can get any mix that we want. Great. Seasonality, dormancy, dormant solves the dormancy. These things aren't dormant. They will grow as soon as you want them to grow. And so some plants that's particularly annoying, especially desert species, looking at you, Southern Africa, because you got some really weird shit down there that takes way too long for the seeds to sprout. And yeah, we don't have that problem with this. So they'll sprout as soon as we want them to. Low seed production also sucks. I mean, especially for old growth, like big trees. Those are super cool trees to have when you're reforesting, but they just barely make seeds, and they're so not usable. And this way, if you got the parameters right, every kind of plant seed would cost the same. Like, it's a little bit of differences in hormones and time, but effectively, like, it's just the cost of the media and the, machining to do it. The plant will just keep growing on its own. So it gives you access to a lot of plants that you'd never actually be able to grow in quantity. The other thing is actually, although it doesn't have a dormancy, and you would think, okay, these are only good for like a month or two, you can freeze them. You can freeze them for 20 years in a deep freezer and just pull them out.
Yeah,
you can freeze the
sick.
you can freeze the liquid culture, which means that I can just have a batch frozen of liquid culture, take out a little bit, and use it to grow out a new liter every time I need some. So I can easily have a long-term storage of these. It's like so flexible. So a lot of people, a smart question is like, what about genetic diversity? Because like it's a bunch of clones. It's not great to plant up a bunch of clones. And it's a great question because it's true. It's not good to plant a bunch of clones. And what should always be aiming for is getting away from monocultures. So how is this better than planting seeds, which you would expect to be much more diverse? Well, the answer is that for most plants, especially wild plants, you're not going to have that much diversity for the stuff that you have in culture. You, are only getting the ones that you can get. So like only the ones that do produce enough seed for you to do. The lines that a lot of people have for this, they're not often going back to the wild and getting new seed stock. They're like continuing the ones they have, either by cutting. I mean, this is how you make enough plants to do this.
This is why apple trees kind of suck, actually.
Man, but I want to go to Kazakhstan. I want to go apple hiking and just like eat a bunch of terrible and some delicious ones.
Hey, we've got 3 crabapple trees that are apples the size of a plum, and they are the best fucking apples I have ever had.
Wow. Wait, are they sour or normal
A
apple?
little bit, but sour is good. Anywho, yes. Sorry.
All right, cool. So yeah, genetic diversity. So basically, with this method, we can go. I'm going to use Java, which is an island in Indonesia, because that's what in Borneo. So in Borneo, if I want to get this ironwood tree, okay, normally I would have like gone and gotten some seeds when it was in season, and then we use those forever. In this way, I can go out to 3 different parts of the island, get some good samples of the ironwood trees in that area, and bring them back. I now have really diverse ironwood from the same species across vast areas. So we can just use that and spread them out when we're planting. And we can even go a step farther. We can like analyze with little markers on the DNA which particular varieties are the most different, and we can make sure that we're using those. I mean, this is how you do conservation for animals that are dying out. You figure out like which ones can breed and have the least fucked up babies. So we can do better than seeds, especially when you're dealing with hard species. Like it's just not even a contest. Yeah. Also, this is cool for agro. Usually native plants are good, but you can test them for stuff like abiotic stressors. Are they a little drought resistant? Are they resistant to fungal pathogens? I can just test this on the cells while they're in suspension culture. And I can just have basically a selection process that I'm doing this for my vanilla right now. We're doing this whole process for vanilla orchids. And one of the things I want to screen against, it's not in the immediate goal, but like by next year, we should have a fusarium resistance in it, just because like the cells that can't handle fusarium extract just die, and only the good ones keep going. So it's a cool way to select for like resistance on something. And last thing is cost. Like I said, super cheap. I mean, it's just so cheap. If you have this machine and you're pumping out syn seeds, the cost to maintain it, to continuously do that, compared to would it be any other method to produce plants, is just an order of magnitude of difference. Once it's set up and you have your parameters for your species, like just keep it going. You just keep it going and you spread what those are. Yeah. Okay. That's it, I think.
Amazing.
So we had a brief conversation about using oh God, and then he died.
Sorry.
No, he's still there. Okay.
I'm here.
We had a brief conversation about more about the process of doing this, and so we were kind of talking about molecular gastronomy and the addition of chitin. Do you want to dive into that a little bit, or do people just?
Yeah.
Does anyone have any questions?
I do.
Yeah.
Hey Diego. I jumped in right as you were talking about the step between callus and somatic embryo, as we were just talking about yesterday.
Yes.
But when you're talking about like the synthetic seed and the main contamination issue being like just the sugary matrix around it, I mean, is the matrix just like MS and sugar and I don't know, like Gamberg's vitamins, or like
at its core, the matrix is usually like some MS sugar thing. The important part that triggers these different events is the hormonal levels. Like plants will have different requirements for their nutrient media, but like you tend to stick with like some kind of MS and sugar. Like that's like the basic, because the sugar is giving it the carbon that it's not getting from photosynthesis or any other way. And the media is just like,
who aren't in the plant growing business. What is MS?
Oh, I get to say that great word too. Muscarage and skoog. That's a good. Yeah, muscarage. I can't even pronounce it.
It's Mursheeg and Skoog.
Mursheeg? That's actually quite beautiful.
Mursheeg.
Yeah. Great.
Yeah, it's Mursheeg and Skoog with Gamborg vitamins.
Camphor, that's awesome. Yeah. Oh, they were. They were the people that figured out what plants needed in agar because nobody could figure it out for a really long time. They're like,
It's the Rondo.
they tried. They what?
It's Brondo.
Great.
It's Blondo.
Also, apparently I read up a little bit on sin seeds yesterday, just so I wasn't coming in here blind. Apparently, Murashig actually made some sin seeds in the 70s.
Yeah, man, they've been
Of
around
course
for
he
like
fucking
15
did.
years. Yeah, it's nuts. And it's weird. The Cincy's like, I don't know why, but just like having come up. There's a really cool paper from the early 90s where they were making sin seeds in like the gel caps that you use for powders and medicine. They just like stuffed them in there.
We have the.
Yeah, it's super cool. It's been around
So
for a while.
other questions. We're still doing Journal Club, people. Other questions.
I got a question.
Yeah.
It's on somatic mutations when you have a liquid culture of plants. Is there any worry about that?
Like somoclonal variation as the cells start to multiply?
Yes, like apparently I've read some books and papers, and it's quite useful that when you grow them in liquid, they just start mutating in weird ways.
Oh yeah.
Like chimeric issues?
No, not chimeric, just like changes. You know, they'll start to like drift and change genetically. This comes up a lot, specifically for agriculture, because the purpose of this, in most of the cases, is to make a lot of the same plants to plant in a giant field or a forest that you're going to get wood from. So you don't want variation in that. And to stop that, you do just genetic screening periodically. You know, you find the genetic markers for exactly what you want to grow, and then when they deviate from that, you're like, no, that's not what we want. So it's really bad if you're trying to get a whole bunch of the same stuff. But somoclonal variation is actually super cool. It's like built-in genetic diversity. Like it's another point against that. Like you can't just plant a bunch of clones because they'll just vary on their own. Now, will some of the variations be not as advantageous and kind of fuck it up? Yeah, sometimes that can happen. But the stage, a lot of the metabolic, if you're getting like a really bad fuck up in the variation genetically, you're going to usually see that in the stage where you're like looking at the little embryos starting to grow. They'll grow like slower or like a little weird. And if you're using the robotic camera eye method, you know, there's ways to do a pretty good selection against that. If you're super anal about it, you can do genetic screening. But I would probably just like, fuck it. Like sometimes seeds don't work, you know, like as long as the rate isn't absurdly high, you're still coming out on top. And the genetic variation, good for us. That's what we want.
Yeah, I got it. But that's gonna be complicated for putting it into like environment. But yeah.
You mean it's complicated for the environment?
Yeah, it's worrying for the agencies that you have to ask if you want to do any.
Oh no, but I don't care. Yeah, but I don't care about that though. So I'll just just plant them anyways. So.
Yeah, well, outside of Europe, yeah, but in Europe, oof.
Okay.
Yeah, and another question is for Gabriel and you. Like, I remember one of the live streams that Gabe was showing basil seeds growing with genetically engineered agrobacterium, and that you were trying if that work for doing the modification. Can you do that with sin seeds?
Oh yeah, you definitely can. I mean, all those little cells floating around in that smoothie, they're just so easy to hit with agrobacterium.
Yeah,
you just squirt
waiting for it.
Yeah.
Yeah. On the other hand, one of the interesting things that I've noticed about the Ent modification is the agrobacterium are super aggressive. And so what happens is that you actually have to, so we've had you actually have to move back and forth between the modification, because if the agrobacterium continuously modify indefinitely, which they will, because that's what they do, they're just like little, yes, more people showing their cats. Let's keep this going.
It's a Bali street dog.
Oh, looks like a cat, and I'll take it. If the agrobacterium just continuously modify indefinitely, they kind of overload the plant and it breaks. So you have to actually sterilize. So the method that I've been using is with seeds, and you have to clean the seeds before you grow them, because if there's agrobacterium on them, it just keeps modifying. It won't stop. So,
Hmm.
And it just inserts the gene randomly too, right? Like
yeah,
you're not guided by anything in
it
this.
just keeps cramming the gene. Yeah. Hey, Louis.
Nice.
That's Louis Pasteur.
Aw.
Pistachio cat. But yeah, so they just continuously modify, and they do it randomly, and like plants are very robust. Diego has already talked about how plants are like super structurally sound. Like they'll just keep doing shit. Like you take a plant, you cram it into the dirt, and plant's like, you know what? Maybe I'll just be a more plant. Maybe it's time to be roots. Whereas there's a finite number of genes that you can cram into an organism before it freaks the fuck out. And so, fox face.
Yeah.
And so,
also with plants, like as long as you've got the right hormone concentration, they're very happy to grow in culture. I mean, the caveat to that is it does actually take a fair bit of time to figure out the exact hormone concentration for each plant, because not only is it different, like this is not the sort of thing you can look up a recipe for, because every plant is very, very different.
Yeah.
And even different strains of the same plant. So like I was trying to do callus culture with mint, and we were following a protocol out of a paper, and it was a slightly different variety of mint, and so the hormones just didn't work. Like they, instead of making nice healthy cultures, like it looked good at first, and then it just got really weird and broke. So what you end up having to do when you're starting with a new plant, you've got the first thing that you do is you essentially set up like 20 plates, and figure out what is the exact hormone concentration that they need. And then from there, it's much easier. Like once you've got it, then things are easy. You just got to make sure you're working really sterilely. But also you kind of have to do this for every step. So like the step where you go from like plant booger to plant embryo booger, also you've got to like you switch out the hormones or like switch out the levels. But you may end up having to try this many times before you find the right mix. And sometimes like plants can be really picky. Like there's there's different classes of plant hormones, like there's auxins and gibberellins and all these different things. But it's not like one hormone. So like in the gibberellic acid category or the auxin category, there's 10 different things that you can buy. And some plants.
Yeah. Right. I also work with plants, so I know how complicated the whole hormone part is. So what I was thinking, what I had in mind is that you have some standardized synthetic seeds that you can modify with a simple protocol with agrobacterium that can end up with much better results. Like you don't have to bother with the whole vegetal hormones. You just get the seed, put it into agrobacterium.
Yeah, I mean that would kind of work. I mean, if you're gonna do that, just do it with normal seeds, or like something like that. And like I think the synthetic seeds only kind of work if they're like,
but
they're fun. They're fun to make at your house and stuff, but it's not like you're gonna make enough that you shouldn't have just bought seeds unless you're really going for it.
But that's what we went to current.
We're going like I'm down. Let's get a wave bag, and then let's do some molecular gastronomy synth seed stuff.
Yeah. I want to try the stuff.
How can we?
As long as I don't need to peel any more seed coats, then I'm happy. Because at the Florida lab, I peeled so many seed coats.
Okay, so you're Nico, right?
Me?
Yeah.
I'm the Nico you talk. Yeah, I'm one of the many Nicos, but yeah.
Dude, the method that I've been working on requires micro germination with no seed coat peeling, because pistachio peeled so many goddamn seed coats, and and now I found this method where you don't need to peel the seed coat. You let the seed coat peel itself.
Yeah, and then my entire fucking refrigerator got infested with agrobacterium after the power to my unit got shut off.
Okay, so that is
Wait, how did that happen?
Because our basic idea was that we would take a 50 milliliter Falcon tube and just put a shit ton of seeds in there with our solution of like really aggressive agrobacteria. And then after a while, I just left one night, and the unit decided that they were going to shut off the power in the middle of the night for some fucking reason. And I come in, and it just smells terrible. And I had to douse the entire thing in isopropyl, and was afraid I was going to start a fire or some shit. It was bad. It was really bad.
Yeah.
And it's Florida too.
Oh, that sounds fun.
Amazon UVC light. Get it?
Yeah. Oh, those are great.
Yeah.
Also, your first mistake was being in Florida.
Yeah.
We get it, Justin. I don't have any choice.
The coolest time I ever had in Florida was like train hopping out of Florida to Georgia. That was really exciting.
God, that seems like my only method of getting out of here right now. You know, in the cesspool that currently is Jacksonville. But
I don't know.
Oh God, minus extra 10 points for Jacksonville.
Yeah.
Hey, that's where I train hopped also.
Yeah.
dude, literally, I was watching this
90% trained.
I was watching this like crime psychology YouTube channel on like just some random channel, and I started scrolling through the videos, and every single time it would be like criminal in Jacksonville, Florida, and I'm like, oh, we're known for something now. Yay!
I don't know.
And they're all like murderers and serial killers. It's great.
Back to the paper, folks. Anyone
Thank
got
you.
any more questions?
Yeah, like okay, how could we engineer around the whole like synseed getting contaminated? Could we just put like a capsule around it?
Well, that is how you do it. You have the capsule around it, but the problem is, so I posted a little while ago. You can look up synseed stages in germination. Essentially, until that capsule ruptures, you're good. Like it's fine. The problem is once the plant breaks through that shell and it's exposed, the bacteria are going to have at it, and sometimes the plant survives, sometimes it doesn't. I think the successful ratio is something like 35 to 45% of them work with bacteria being a problem. But there are things. I mean, you're talking about China.
The addition of chitosan.
Mm-hmm.
Yeah, well, there's chitosan, but then you can
actually reduce. Yeah.
Well, there's
actually. It reduces the amount of contamination. It's antibacterial. You can mix it in.
Yeah. Do you know if you're using chitosan or are you using the chitin B deacylated coating?
No, it's not the deacylated. It's just chitosan. You mix it in with your calcium chloride and your alginate. Did I get those chemicals right? Hopefully, yes.
Kind of, yeah.
Yeah, and you just and you do that. Didn't I find you a paper about that? I have definitely put it in the library.
You just put it in the calcium, I think, so that way when the alginate hits it and cross links and you pull it out, it's got like a chitosan coating on the outside and then alginate on the inside.
Yeah. So this thing is in the library. It exists. Once
you could also throw like a shellac coating on top just to really seal it in there, because the seed's gonna burst out anyway. It's not gonna matter.
Right.
Like,
well, so, I mean, so when we were in Chile, Justin, remember when we were in Chile, all of the seeds were covered in clay. They were like these weird little clay beads. And so there's also this other concept where we can just take all these synthetic seeds and cover them in dirt. We're basically seed bombing, but with GMOs.
I mean, that's literally what.
Did the chito caps that I made ever end up working, or did the seeds just die?
They died.
Damn it!
I mean, Gabe, at the company I'm at now, like that's literally like we did this a month and a half ago. Like we took some alginate and coated seeds, and then we put a capsule around it, and then we literally just took like fine sieved compost and clay, and then we just encrusted it, and we were trying to just create a delay fuse on.
Yeah, you made a seed bomb.
Yeah, it was a seed bomb. It was a delayed fused synseed.
Yeah, it was a delayed fused synseed. Yeah, it was a delayed fused synseed. Yeah, it was a delayed fused synseed. Yeah, it was a delayed fused synseed. Yeah, it was a delayed fused synseed. Yeah, it was a delayed fused synseed. Yeah, it was a delayed fused synseed. Yeah, it was a delayed fused synseed. Yeah, it was a delayed fused synseed. Yeah, it was a delayed fused synseed. Yeah, it was a delayed fused synseed. Yeah, it was a delayed fused synseed. Yeah, it was a delayed fused synseed. Yeah, it was a delayed fused synseed. Yeah, it was a delayed fused synseed. Yeah, it was a delayed fused synseed. Yeah, it was a delayed fused synseed. Yeah, it was a delayed fused synseed. Yeah, it was a delayed fused synseed. Yeah, it was a delayed fused synseed. Yeah, it was a delayed fused synseed. Yeah, it was a delayed fused synseed. Yeah, it was a delayed fused synseed. Yeah, it was a delayed fused synseed. Yeah,
Yeah, I mean, so like you can put stuff around the sin seeds for sure. I mean, that makes a lot of sense. You're protecting it from the elements. And in fact, if you're looking at the aerial reforestation drones in the Northwest, they use charcoal around the sin seeds. Tends to work really good because it also protects it from the sun, which is important because they can just get too hot and that kills them. But so one option is just an antimicrobial like chitosan, like you guys were talking about. But another option, which I think is really promising, would be either in the sin seed itself or in the casing around it, introducing some rhizomorphic bacteria that form, they form relationships with the roots of these plants. And not only do they help with abiotic stressors like drought and finding nutrients, but they also actively excrete metabolites to protect the plant.
Right.
Those metabolites are really helpful.
Right. So this is a really good idea, but it's also problematic. So I call this the fishbowl problem. In as much as that the smaller the system that you're working with, the more likely it is to get unbalanced. So like a goldfish bowl, a little bit of pH shift, and holy shit, if that thing doesn't get toxic. Whereas with like a 20-gallon tank, a little pH shift doesn't mean the same thing. And so the rate of growth, the requirements, and I mean, again, even the pH levels for adding other organisms into one condensed nodule generally tends to lend itself to things moving at different rates. And so if you want to get super nerdy about it, if you've ever read any books about colony ships, like going into space or something like that, there's this problem where the bacteria on the ship evolve faster than everything else. And so you get weird issues with spoilage. And so, yeah, it's a thing. Sci-fi, it's fun. Also has great ideas. These people do their research. And so what you end up getting is that if you try to cram too many species into a small place, some of them will freak out. So having a multitude, a varietal of things and letting them work together usually tends to be better than trying to cram everything in. Like, don't make one thing try to do all the things. Like,
yeah, the main thing that we're trying to do is just stop them from getting infected by bacteria. So like there's abiotic ways to do that, but surely even for agriculturally, you can buy a spore inoculate with I think 10 to 14 species of mycorrhizal fungus, and use that to inoculate. So like, I don't know how much it would affect that. What if you sprinkled that in the soil around it?
That works really. Yeah, I was gonna say that works really well if you're sprinkling that on soil. It doesn't work so much if you were to inject it into the seeds, which is basically.
No. I mean, like in the coating, like around it, the soil.
Well, yeah, but that. Okay, well, see, those are two different things. When you say in the coating, that implies like in the sin seed, whereas if you say in the soil around it, okay.
You're right. I need a better word for that. Whatever, like the stuff. You have the sin seed, and then you're going to have like soil or peat or something around it. You're not just going to have a naked sin seed, so whatever that's called.
I mean, the other option is you go something like colloidal silver or something. You mix that into the seed, the sin seed, and that'll keep everything else out. But I mean, it'll nuke everything else. Is the problem?
A question.
Yeah.
I don't know if this applies at all, but I was talking to a brewer, and one of the ways that they kept their tanks of beer from getting contaminated during the fermentation process is they would basically like lay a layer of CO2 on top of their fermentation tanks, so that it wasn't exposed to oxygen during overnight when nobody was in the brewery. So like, if instead of trying to include something like colloidal silver or organisms or molecules that are going to continue to live at different rates than the seeds, would it be possible to basically just like keep it in a container with a neutral gas or something that's not going to be metabolized by other thing to keep it from spoiling? Argon? Like, is this a completely off the wall idea?
Nitrogen is what we use for biology, actually.
It just depends what you're using them for.
Nitrogen is like natural gas.
You'd also probably asphyxiate the seed, though. That's my biggest concern with that. Like, it works with brewing because like yeast are fine in basically pure CO2, but plants need to breathe. So if you did that, like everything would die potentially. I mean, it depends on how fast the gases diffuse, but I don't think there's going to be a lot of oxygen diffusing in at a baseline. So cutting it off entirely, I think everything just dies.
Different gas that's only going to, I mean, like fermentation, you know, it's just like creating the conditions so that only the thing you want can live. Like, is there something we can do with that? Do you think?
Yeah, I mean, fermentation is cool, but it is a limiting factor. I mean, by fermentation, either by increasing the pH or increasing some other factor, you're excluding a lot of organisms, and that's great for the organisms doing it. But in this particular case, like if you're going to increase the pH or something of the soil, that's going to affect your plant, even though it would effectively clear the area maybe of a lot of bacteria. But it's also important to just differentiate, like the syn seeds themselves are fine. It's like after they start sprouting then. So, you know, how are you going to keep gas on it while it's sprouting or, you know, in the soil?
I guess I was just thinking about storage or transportation during certain parts of the cycle, you know? So, I mean, like after it's in the ground, obviously all bets are off. But if you're still trying to like grow your seeds, then like it'd be pretty easy to just like control the air content. And I'm just thinking about reducing the growth rates of everything.
They store good. Just freeze them. Just stick them in a freezer.
And
take them out when you use them.
the research on using chitosan is probably the best idea.
Well, also, like, is the issue just like the little remnants of synseed that are left over, and like the little microbes are like, ooh, yummy MS and sugar, we gonna eat that, and then
Yeah, the media that's left over, they eat that, and they're all around
I mean,
the plant.
can you not just calculate like a minimum viable amount of media for the synseed, and then have some sort of coding so that you can like have some sort of calculation of, okay, if we have MS, here's the minimum of MS we need for like, I don't know, the first 4 weeks, 5 weeks, 6 weeks of growth. We got our little taproot. MS is gone. It's all used. We have a little bit of clay coating just to protect, you know, in the meantime, that lasts 4 or 5 months. Then the taproot comes out and, hey guys, all of the sugar's gone. Fuck you. Go get your sugar somewhere else.
I think the problem is that the bacteria are going to get in that first couple of days. It's not going to be weeks.
Yeah,
Like
but if you have a coating around it,
yeah, but if you've got motile bacteria, like the second that plant pokes out of that coating, it's free for all. Like, I think that really the issue is that plants don't have much of an immune system until they've grown a little more. So like they got to get big, and so like I don't think that there's an easy solution to this other than, you know, try the chitosan thing to kind of stave off the infection for as long as possible. But like I think the assumption is you're gonna lose like 30-40% of these when you plant them. Like,
Just make a lot of them, you know.
Yeah. I mean that's the whole point, right? That's what we started with, is that you can just make a just ridiculous shit ton of seeds. So you plant the way that an oak tree plants. You don't care.
Would there be any way to induce like a tougher, woodier callus on the outside through the agro modification?
But all those things come as those things come as a cost, though. Like by doing that, you're gonna fuck something else up.
But also aggro.
This is the thing, Stash. When you make it be the thing, it becomes the thing.
But I mean, you can do that chemically.
What the fuck does that even mean?
Like in your coating formulation, you could do something that is more sturdy, which is why I said something like shellac. So you do like alginate chitosan shellac. Like you have all those the extra layers to you know, increase stability and that sort of thing. But like past the point, you kind of
shellac is not going to be friendly.
Did somebody say argonite coating on the outside? Because that's what I heard. That's a terrible idea.
Lignin, like plants, like trees, they use lignin as a like they defend themselves with lignin. How about we introduce some lignin in the coating?
Because lignin isn't soluble. Like there's no way to like. That's just like if we put some sawdust in the coating, like that's not gonna do it. If anything, it's gonna because the thing is lignin is also food. Like if we were you know, in that like nice 50 million years between like lignin was invented and fungi just figured out how to eat it, like then yeah, sure, that would work great. But we're not, and so there's lots of fungi that eat lignin. So it's not actually great. Also, I think
we could just slam the paper from last journal session to make bacterial precipitated what is it, carbon or calcium carbonate.
I mean, that's harder. I mean, like, yeah, maybe you could use the spider silk goo and just like, dip them in some calcium before.
Let's use chitosan and spider silk, and let's just mush all the projects into one.
Okay.
That would be the coolest thing.
This has been a consistent issue, and it's also been part of why sorting the library has gotten so weird. Does anyone have any more questions about the paper?
Let me say one last thing on the subject of the synseed contamination, because there's one other option we didn't discuss, and that is what's getting contaminated. It's the sugar. So the somatic embryos need that sugar, but once the cotyledons come out, it doesn't technically need it. It can be photoautotrophic. So I think I saw some paper. I don't know sure if it was a fever dream, but where they had like a double coating, and the first coating was sugar, and then it broke through that one into not carbon-free medium, something like that. And basically the outer coating didn't have the sugar problem, and it was trying to be photoautotrophic at that point. And then, you wouldn't have the sugar. It's just making its own sugar. But this has some logistical problems for like planting in mass, but I guess theoretically it works.
What you could maybe do is, starch. Starch is another problem, doesn't it? I was going to say, coat it in starch on the outside, and then put an enzyme in there to break the starch down into sugar. So it's the same way a real seed works, or at least malt.
I mean, starch still contaminates, but I guess a little less than sugar would.
So basically, we're making seed dumplings now.
Sure, why not?
We're basically making seed dumplings.
And now's the Taste Emporium crossover.
Into the alginate, and then we roll it in cornstarch.
Deep fry them. Yeah, I got it.
Gobstoppers.
Popcorn.
What is the heat tolerance of the calluses?
No.
Absolutely not.
But have you seen the cool?
Funny, they get really upset.
But have you seen the heat forming for the Kaido dipping? Like there was this one company that did the basically made a metal cup mold and then just dipped it in Kaido until it formed enough layers to be protective. So you could try something similar with low heat, having like a chamber that would heat it up as you dip the calluses in.
What I was thinking, based off of some of the work that Pistachio and I did in the past, is you would make your alginate globes, and like I said, we did find that paper where they had mixed the chitosan in, but we're also talking about making shells. Chitosan is a shell. So what if we make the alginate globes, and then we basically just kind of eat them all into a vat of chitosan and pull them out really fast so that they get coated, and then let them kind of air dry.
Do you remember the tiny beads that I made from the chitosan sodium hydroxide mixture?
Yeah.
Yeah, you could just like put a large amount of chitosan around it, dip it in the sodium hydroxide, and then leave it to dry for a while. And I don't think that would end up harming the internals.
What I was thinking, what if you use the chitosan instead of agar for the media? Doesn't it thicken it in the same way agar does?
Yeah, but it doesn't cross-link though. So the reason everyone uses alginate is because the second that shit hits calcium, it instantly hardens.
Yeah, so it doesn't cross-link, and secondly, chitosan needs an acid to become soluble. And
a weak acid.
And
but still, it's enough to throw off the
pH,
and the plants are going to be unhappy.
No, but it's like it's usually 5.8. Like it's still an acidic solution most of the time when you're doing
4% vinegar is not seed friendly.
There is a.
It's not bio friendly.
There is a hydrogel hybrid that's chitin and alginate.
The pH of vinegar is about 2.5, just for context. That's plants die.
Is that what it needs? It needs a 2.5?
Yeah.
Okay.
And especially if you're going to like concentrate the chitosan down so that you've got a higher concentration, like pH is going to drop further.
Cool. So this sounds like stuff that should be discussed in the channel. Does anyone have any? This is Journal Club. Is there anything left for Journal Club?
I have a question.
Yes.
So in the beginning of the paper, it says, embryos develop with good connection between root and shoot and without plagiotrophism, often associated with propagation by cuttings. What does, yeah.
I mean, basically, I actually don't know what that cool word means, but I do know what the issue that it's talking about, which is basically, since we're starting from basically a single cell, like a seed normally would, the whole plant develops as a plant normally would. The roots are a continuous part of the stem and the leaves, and it's all like working together. If you're doing like organogenesis or a few other, or like from cuttings and stuff, like you put a stick in the ground and then that stick starts to grow roots, and cool, it'll grow, but like the plant's gonna look kind of weird. It's gonna have like a big old stick with a little root. And so in some plants and in some particular situations, this is bad. It's just not a well balanced plant. So we kind of saw. Yeah.
Sorry. So I looked up plagiotropism. It's the tendency for tropism, i.e., growth response of a plant, to be oriented at an angle to the line of action of the stimulus concerned. So if it's so like stimulus according to gravity, it grows up, and everything is oriented accordingly, basically.
Yeah, that makes
sense.
sense.
It's basically what that sentence is saying is plant grows properly, as if it was a real seed, is essentially what that's trying to say, but using like the $10 words because it's a paper and they can.
It's a good paper. They can use a $10 word sometimes.
Oh yeah, but it's just to explain what that is.
Yeah.
Also, just a hint for when you're reading papers, especially if you're new to this. If there's a word you don't know, just look it up. Like Google is your friend. There's a lot, especially if you're reading something in a field that you're not comfortable with. Just be prepared to be Googling shit constantly, because every time you learn a new word, it makes more and more chunks of the paper make sense.
Or you can do the alternative, which is just make up words for the other words, because it doesn't actually
Don't
matter.
do that. Fuck biologists.
No, it doesn't actually matter if you understand 16 syllables of chemistry.
But you get to say them. Come on, saying the word is so fun, and especially when it sounds cool.
Okay, I'm going to send you an organic chemistry paper, and you're going to take that back.
No, that's why I didn't. That's why I didn't do academia.
Does anybody want to learn organic chemistry? No, you don't. Except for Bast, evidently. She's weird. But the rest of you, just throw in a placeholder. And if there's a word that looks like it is important, definitely Google it, because you should know how the word works, but you don't have to remember the word or quote unquote learn the word. You just need to know that it does the thing. It doesn't really matter.
That's literally the definition of what learning means.
Yeah.
I'm loving the Gabriel Justin interactions on this call. It's making my day.
Yeah. Wow.
All right. You literally were just like, don't learn the words, just learn the word.
What? No, you just learn what the word means, man.
You just learn what the word means, and then you make up a new word, like gobbledygook, instead of
gobbledygook.
No, you learn the word.
And then I just put 50 schmeckles. I just put 50 schmeckles in her hand, and
Yeah, don't use plasiotropism. Just call it schmeckalotropism, because that makes it better for some reason.
Actually. Okay.
That sounds important in a different language.
Coders that are here.
Adorable.
Coders that are here, we already talked about this. You guys know the concept of foo and bar. You know what coders actually. You understand? Sometimes you have the foo, and the foo interacts with the bar. That's it. You don't need to know this shit.
Yeah.
There you go. Oh, it's
Also, the similarity of Diego and Justin's voice on this call has been very funny to me as well. I'm having trouble figuring out who's talking sometimes.
We have
You know,
exactly the same
really?
haircut style too. It's it's kind of annoying there.
Are you guys both from Canada? Is that the reason this is happening, or what?
No, man, I'm Cuban, and I've just lived in like the jungle for 8 years, so my accent is just a weird me thing.
Jeez, it's strange.
So I guess I'm just not saying a boot.
Sorry.
I know they don't sound anything like each other.
Yeah, you just don't hear me going, oh fuck, buddy.
Oh God, jeez, don't do
That's
that
your,
again.
Give your balls a
that's
cut.
your loss.
Okay, wait, I'll be Gabe. Paper stuff. Paper.
I
Okay,
think
go.
we've cut, is there any other paper stuff? I think we're done. Like,
I did have one question paper wise. Was this at all along the backdrop of the structure thing we were talking about, Diego, or is this just paper talk?
I mean, when you got somatic embryos, you can do all kinds of crazy stuff, including like putting them in a very dense gel and then trying to induce them to all form a woody callus into one another. And if you're using something like a ficus that already likes alloplasty and likes binding to itself, then I don't know, maybe you'll end up with a nice continuous sheet of living structure. Like that would be really fun. I've been trying to do that for years.
I would like you guys to know that if you guys were talking about human beings and nobody actually understood the words that you're using, this would be terrifying.
And then I made a sheet out of testicles.
I am so happy. You know, you just like make some somatic embryos and you can just implant them anywhere.
I'm glad we're in your digital house then.
Yeah.
So, are we starting another channel for Human Centipede? Is that what I'm hearing? No? Okay.
Just sheets of testicle.
Henrietta Lacks doesn't have shit on us.
And nobody can ask why we're doing it. It's just like, what are you talking about?
Yeah, rule number one of Bio Club is you don't ask why.
Actually, rule number one is you don't ask where the materials came from.
Did somebody ask me berries? Did somebody say me berries? That's what I heard. I don't know. Yeah, Justin.
I get so many fucking comments. Like, I wish we'd done it. I really wish we'd done the me berry.
I thought it was going to work.
Well, we just didn't have the stem cell kit. That was the problem. If we had the stem cell kit, we totally could have done me berry. Although, like, thinking about it, we could have just taken like a muscle sample or something. Like,
wait, you never.
I am happy that like the meat berries from your actual channel did not make it onto your cooking channel, Justin.
Yet.
No, that's definitely where that would have gone, because
You should have figured out how to make it like differentiate into testicle skin tissue. Then boom.
Wow, there's a bright future ahead of us.
On that note,
I'm
doing stupid shit. So
okay, so I think Journal Club is done.
Thank you for coming to Journal Club, everyone.
It was good talking to everybody.